Imagine going hours without eating. How does your body keep your blood sugar in check? What happens when you enjoy a meal packed with carbs and sugars? All of this is carefully managed by a group of hormones that work behind the scenes, ensuring your blood glucose levels remain stable. But how exactly does this process unfold? In today's video, we'll explore the key hormones that regulate blood glucose, how your body responds to fasting and feeding, and what happens when this system breaks down. Let's dive into the fascinating world of hormones, insulin, and glucose regulation. When you're fasting, whether it's overnight or skipping a meal, your body needs to keep your blood sugar levels steady. Here's how it works. The liver steps in by breaking down glycogen, a stored form of glucose, and releases it into the bloodstream to give you energy. At the same time, your liver and kidneys start a process called gluconeogenesis, where they actually produce new glucose from non-carbohydrate sources. This means your body can maintain blood glucose levels even when you're not eating. So, even when you think there's no food coming in, your body is already working to keep everything in balance. Now, let's talk about what happens right after a meal. When you eat, the carbohydrates from your food are broken down into glucose, which enters your bloodstream, causing a spike in blood sugar levels. Your body then stores some of this glucose as glycogen in the liver and muscles. Any excess glucose? It's stored as fat in adipose tissue. This helps to quickly normalize your blood sugar after eating and provides energy reserves for later. Hormones that regulate blood sugar levels. The key player here is insulin, but there are other important hormones too. Insulin, this hormone, produced by the pancreas, lowers blood sugar by promoting the uptake of glucose into muscles and fat cells, suppressing the liver's production of glucose. Counterregulatory hormones, on the flip side, hormones like glucagon, epinephrine, cortisol, and growth hormone raise blood sugar when it gets too low. They work by stimulating the release of glucose from stored glycogen, increasing glucose production in the liver. Together, these hormones ensure that your blood sugar levels stay within a healthy range, regardless of whether you've eaten or not. Let's focus on how various organs respond to insulin. Liver, muscles, and fat cells, these are the primary organs that respond to insulin. Insulin stimulates glucose uptake into muscles and fat cells via a special transporter called GLUT4. Interestingly, insulin does not promote glucose uptake in liver cells directly. Instead, it focuses on suppressing glucose production in the liver. This coordinated effort ensures that glucose is used efficiently across the body. For your body to regulate glucose properly, a few things need to happen. 1. Pancreatic function. The pancreas must be able to produce and release insulin effectively. 2. Glucose uptake. Insulin needs to promote glucose uptake into muscles and fat cells. 3. Liver control. Insulin must suppress glucose production in the liver to prevent blood sugar from rising too high. When any of these factors are out of balance, it can lead to issues like diabetes. Let's dive deeper into insulin, the star of blood sugar regulation. Production. Insulin is produced by the beta cells of the pancreas in response to rising blood glucose levels. Function. Insulin is an anabolic hormone, meaning it helps build up energy stores by stimulating glucose uptake into muscles and fat, storing glucose as glycogen or fat, inhibiting glucose production in the liver. Synthesis. Composed of 51 amino acids in two chains, A and B, connected by disulfide bridges. Insulin starts as pre-proinsulin, which is converted to proinsulin, and finally cleaved into insulin and C-peptide. Release. Insulin is released in two phases. Phase 1, a rapid release of stored insulin within the first 1 to 2 minutes. Phase 2, a slower, sustained release that lasts up to 2 hours as your body continues producing more insulin. Insulin is also tightly regulated by factors like incretin hormones, GLP-1 and GIP, and gut hormones, which enhance its secretion in response to food. Degradation. around 50% of insulin is broken down in the liver after being released into the bloodstream. Additional degradation occurs in the kidneys, where insulin is filtered, reabsorbed, and broken down. Insulin's half-life in the body is about 4 to 5 minutes. Insulin in health and disease. In healthy individuals, insulin is released in a pulsatile manner. However, in certain conditions, things can go awry. In type 2 diabetes, the first phase of insulin release is often lost, though the second phase might remain. In type 1 diabetes, there is little to no insulin production at all, leading to a dependence on insulin therapy. Proinsulin and C-peptide. Finally, let's briefly touch on two related molecules. Proinsulin. Proinsulin has about 10% of the biological activity of insulin and serves as the major storage form of insulin. Normally, only a small amount, 3% compared to insulin, of proinsulin is released into the bloodstream. Proinsulin is cleared by the liver at 25% the rate of insulin, with a longer half-life of about 30 minutes. In the fasting state, proinsulin levels in the blood are around 10 to 15% of insulin levels. C-peptide. Proinsulin is split into insulin and a 31 amino acid connecting peptide, C-peptide. C-peptide was once thought to be biologically inactive, but newer evidence suggests it may have some biological activity, though its role is still debated. Insulin and C-peptide are released into the blood in equal amounts, but fasting C-peptide levels are 5 to 10 times higher than insulin levels due to its longer half-life, 35 minutes. The liver does not break down C-peptide, instead, it is removed by the kidneys, with some being excreted unchanged in the urine. Mechanism of insulin action. Insulin works by binding to specific receptors on the surface of cells, which then triggers a cascade of events inside the cell. Here's the step-by-step process. Insulin binding. Insulin binds to receptors made up of 2 alpha subunits, outside the cell, and 2 beta subunits, spanning the cell membrane. Activation of the receptor. Once insulin attaches to the alpha subunits, it activates the beta subunits, which have tyrosine kinase activity. Signal transduction. The activated receptor phosphorylates itself and other proteins, including insulin receptor substrate, IRS, proteins, which serve as docking sites for various signaling molecules. Pathway activation. This phosphorylation sets off several pathways, like the MAP kinase cascade, which promotes cell growth, and PI3K ACT signaling, which is crucial for glucose metabolism. One of the key effects of insulin is that it moves GLUT4 glucose transporters to the cell surface in muscle and fat cells, increasing glucose uptake. In the liver, insulin has a different role. It boosts glycogen synthesis, suppresses gluconeogenesis, glucose production, and promotes lipogenesis, fat production. Complexity and redundancy in insulin signaling. Although we understand many aspects of insulin signaling, it remains highly complex. For example, studies in mice have shown that even when key insulin signaling molecules, like IRS-1 or GLUT4, are knocked out, the animals don't always develop diabetes. This suggests that there's redundancy in the system, meaning other pathways can step in to compensate, ensuring glucose levels remain balanced. Glucose transporters, the key players. Glucose enters cells through 2 major families of transporters. 1. Sodium-dependent glucose transporters, SGLTs. These transporters use sodium gradients to move glucose against its concentration gradient. They're found mainly in the small intestine, absorbing glucose from food, and kidneys, reabsorbing glucose from urine. 2. Facilitative glucose transporters, GLUTs. GLUT transporters allow glucose to move across cell membranes following its concentration gradient. There are 14 GLUT transporters, GLUT1, GLUT14, with 11 known to transport glucose. Let's focus on a few key ones. Types of GLUT transporters. GLUT1. Present in many tissues, it ensures a basic level of glucose for most cells. It plays a critical role in the blood-brain barrier in transferring glucose from mother to fetus via the placenta. GLUT2. Found in the liver, pancreas, intestines, and kidneys, GLUT2 has a low affinity but high capacity for glucose. It allows glucose to flow freely in and out of cells, making it the primary glucose transporter in liver cells. GLUT3. Primarily found in neurons, GLUT3 transports glucose to nerve cells where it's essential for brain function. GLUT4. This insulin-responsive transporter is found in skeletal muscle and fat tissue. After eating, insulin signals GLUT4 to move to the cell surface, allowing glucose to enter muscle and fat cells. In people with type 2 diabetes, this process is impaired, which leads to higher blood glucose levels. GLUT9. Though it doesn't transport glucose, GLUT9 plays a role in transporting urate, a key player in conditions like gout. There are other GLUTs like GLUT6, GLUT7, GLUT10, GLUT11, GLUT12, and GLUT14, whose functions are not fully understood yet but were identified through human genome sequencing. Insulin-like growth factors, IGFs. Besides insulin, the body also produces insulin-like growth factors, IGF-1 and IGF-2, which share structural similarities with insulin and have important growth and metabolic effects. IGF-1. This hormone is a key mediator of growth hormone, promoting cell growth and differentiation. It is primarily produced in the liver but acts locally in other tissues. Unlike insulin, most IGF-1 is bound to specific proteins that regulate its activity and protect it in circulation. IGF-2. Although its role is less understood, it's thought to play a part in cancer development. Overproduction of IGF-2 by certain tumors can even cause fasting hypoglycemia, low blood sugar. IGF receptors and clinical importance. IGF-1 receptors, these are similar to insulin receptors and can bind both IGF-1 and IGF-2 with high affinity. IGF-2 receptors, these are quite different. Unlike IGF-1 receptors, they don't have tyrosine kinase activity and don't bind insulin. They bind IGF-2 strongly but are involved in different processes. The clinical importance of IGF-1 is significant. IGF-1 deficiency can lead to growth disorders like Laron syndrome, while excess IGF-1 can cause conditions like acromegaly. Measuring IGF-1 levels is critical in diagnosing growth hormone-related disorders and assessing nutritional status. Counterregulatory hormones are your body's emergency response to low blood sugar. Here's what they do. Opposing insulin, these hormones act against insulin by promoting the breakdown and production of glucose in the liver. Raising glucose levels, they increase blood sugar by stimulating glycogenolysis, the BRI. Are on gluconeogenesis, the creation of glucose. These hormones are critical in maintaining blood sugar balance, especially when glucose levels start to drop. Immediate response to low blood sugar. Your body responds to low blood sugar within minutes, thanks to the rapid action of two key hormones. Glucagon and epinephrine, also known as adrenaline, act fast, boosting glucose production almost immediately. Growth hormone and cortisol take longer. After 3 to 4 hours, they step in to further mobilize glucose and reduce its use by other tissues. This multi-phase response ensures your body has a steady supply of glucose, even in times of stress or fasting. Glucagon, the primary counterregulatory hormone. Let's talk about glucagon, the hormone most responsible for raising blood sugar. What is it? Glucagon is a 29 amino acid hormone secreted by the alpha cells of the pancreas. How does it work? It targets the liver, promoting the breakdown of glycogen into glucose and stimulating glucose production, while also inhibiting glucose storage. Regulation: low blood sugar triggers glucagon secretion, but in people with long-term diabetes, this response weakens, increasing the risk of hypoglycemia. Additional roles: besides glucose regulation, glucagon influences fat metabolism and ketone production, both essential during fasting or low-carb diets. Glucagon also plays a role in the gut. Proglucagon is cleaved into GLP-1, which helps balance blood sugar by stimulating insulin secretion and inhibiting glucagon itself. Epinephrine, adrenaline, the fast-acting backup. Next up is epinephrine, often known as adrenaline. What is it? Epinephrine is secreted by the adrenal medulla and works to quickly raise blood sugar by stimulating glucose production and reducing glucose use in tissues. How does it help? Epinephrine promotes glucagon secretion and inhibits insulin secretion, making it especially important in cases where glucagon is deficient, like in type 1 diabetes. Stress response. When you're under stress, epinephrine surges, releasing energy in the form of glucose. However, tumors like pheochromocytomas can cause excessive epinephrine secretion, leading to hyperglycemia. This hormone acts as an emergency fuel release system, making sure you have energy when you need it most. Growth hormone, slower but effective. Growth hormone takes a bit longer to kick in, but it's still essential in managing low blood sugar. What is it? Secreted by the anterior pituitary, growth hormone enhances glucose production, stimulates fat breakdown, lipolysis, and blocks insulin's effects on glucose uptake. Why is it important? By preventing glucose from being used too quickly, growth hormone ensures a longer-lasting supply of energy. While not as fast-acting as glucagon or epinephrine, growth hormone plays a critical role in long-term glucose management. Finally, there's cortisol, often called the stress hormone. What is it? Cortisol is released by the adrenal cortex in response to ACTH and is essential for boosting glucose production and breaking down proteins and fats. What happens with too much or too little? Excess cortisol, as seen in Cushing's syndrome, can cause hyperglycemia. Cortisol deficiency, as in Addison disease, can lead to hypoglycemia. Cortisol's role is crucial during prolonged stress or fasting, ensuring that the body has enough energy by converting protein and fat into glucose. In addition to the main counterregulatory hormones, several others influence glucose metabolism indirectly. Thyroxine, T4. Secreted by the thyroid gland, thyroxine isn't directly involved in blood sugar regulation but plays a role in glycogen breakdown and increases intestinal glucose absorption. People with thyrotoxicosis, an overactive thyroid, may experience glucose intolerance, although fasting glucose levels usually remain normal. Somatostatin. Also known as growth hormone inhibiting hormone, somatostatin is a 14 amino acid peptide found in the gastrointestinal tract, hypothalamus, and delta cells of the pancreas. It inhibits the release of both glucagon and insulin, acting as a balancing hormone without directly influencing carbohydrate metabolism. These hormones help fine-tune the complex system of glucose regulation, ensuring your body remains in balance. Thank you for completing the video. Don't forget to share, subscribe, and hit that like button. Till next time, bye.